Recording begins. It is November 19th. This is Biology 105. Thank you all for making it to Biology 105. It's so good to see all of you once again. Let's look at the schedule. All right? Look at that. The whole thing fits on a screen. That feels good. Here we are in week 13. You did your exam. Nice job, everybody. Good average scores. I'm thrilled. All right? Your scores should all be put in. If they're not, then let me know because I missed them. Yeah, question.
Yeah, that actually reminded me. I took mine in the testing center. Did you?
I'm picking those up today. I picked up some, but then other folks took them later in the day, and so I'm getting those right after class.
Okay.
Never hurts to clarify that, though, because other people might be in the same boat, so thank you. Yeah, your grade should be in by the end of the day. Otherwise, exam 3 scores uploaded. We're ready to talk about the central dogma, DNA replication, all sorts of stuff. And the remainder of our semester is essentially a genetics course, okay? We're gonna go genetics, and then a little bit about like evolution and natural selection, okay? Question? Well, you're in luck because that's the next thing on the docket. So let's just slide into that discussion right now. At this point, all 3 of your midterms have been completed. This means that all 3 of the assignments that you can do makeup points for are done. That was a horrible sentence that I just structured. Here's the thing. If you got under 70%, which I believe is like a 52, something like that, let's just put up a calculator and figure that out. What's 70% of 75? 52. Nailed it. If you got like a 52 or lower on any of those midterms, come to my office and take your exam, a copy of the exam. You take it and you write a report about each question you missed, okay? 3 to 5 sentences for each question you missed. You explain to me what you answered, why it's incorrect, the correct answer, and why it is correct. You get back half the points that you missed. Takes a failing grade, turns it into almost passing grade, essentially. All right? So if you want more details about that, just drop by my dang office. When are John's office hours? Well, they're right here in the syllabus, okay? Mondays, 12 to 1. Tuesdays and Thursdays, 10 to 10:50 in Alastair McCrone Hall, room 169. Or you can set up an appointment with me, but at this point, there's gonna be a lot of people vying for appointments, so if you can make the office hours, that's great. It takes about 5 minutes for me to give you your materials and send you on your way. You do the report on your own and then drop it off at the end of a lecture to me. Pretty straightforward. Questions about that? Is that solid for you at the back there? Yeah. Cool. Yes?
When's the latest you can get revisions in?
The week before finals week. Yep. I give a deadline of like 2 weeks from when you take your exam, but I'm honestly pretty flexible with that. But I don't want to take any during finals week because I want everybody's grades to be done, by the time you start taking your final. Awesome, yeah.
Do one or all 3?
Just one. One time per semester. So whatever midterm you got the lowest score on, that's the one to do the makeup for. Additional questions about that? Okay, solid. So we're on the course schedule here. A couple more weeks. It's fall break coming up, so let's just wrap up week 13. Keep an eye out for, announcements about the finals. There's a lab final exam during week 16 of the semester, so not during finals week, the week before. That's that open lab notebook, lab final that I've been talking about, the whole time here. So make sure that's on your radar still. Yeah. Question? The online lab, sorry, one more time?
Yeah, online lab.
Is it under the quizzes, assignments? Where's that showing up for you? Modules, thank you very much for letting me know. I'm certain that it's just a holdover. Because on my end, I've got all of these things that are unpublished and published that I swap out depending on semesters. And sometimes I publish something that I mean to have unpublished. It's good to sort that out. Thank you. This is great. Just sitting on a loading screen. Good deal. In week 15? Thank you. Population Genetics Online Lab. Yes, that's actually not what we have going on in week 15. Thank you very much. You actually have the regular old lab right here on population genetics and gel electrophoresis. So there's a regular in-person lab in week 15. Yeah. Trust me, it's something you definitely want a TA to give you a hand on, because it's a pretty intense lab. You do Hardy-Weinberg equilibrium, and if you haven't done that before, it's mentally taxing. Yeah. Yep, right there at week 15. I go back and forth. I tried the online thing, and the problem was everybody failed that question on the lab final. So I was like, well, nobody actually got Hardy-Weinberg, and you need it if you're going on to upper division bio stuff. Anyways, that's neither here nor there. Thank you so much for catching that, because that would have become a problem very quickly. Okay, any other questions? I mean, this is, a good point in the semester to really just finish strong. We want to make sure you've got all the tools. There's also a lab practice final. That's an online assignment the week before your lab final exam. That is essentially a complete study guide for your lab final. So if you're concerned about what's going to be on the lab final, it's all of the topics in the lab practice final, okay? So this is also a study guide resource for you. But I will not be publishing a study guide for you for the lab final, since it is open notebook. Question?
You might have answered this earlier in the semester, but how many pages do you think the lab final is going to be?
Oh gosh, I got it written. Yeah, I honestly can't remember. I think it's like maybe 10 pages, but there's some questions that take up an entire page. Like the Hardy-Weinberg question is a page, of it. I will tell you that it is similar to the last semester's lab final. And that lab final, some people finished it in 30 minutes and got 100%. Some people took the full 3 hours and got 100%. All right? So if that's information that's useful for you. Yes, a question. Your lab notebook can have anything you want in it. If you want to take an entire Wikipedia page and print it out about like how to do Hardy-Weinberg equilibrium, do it. It's totally fine. That's how a lab notebook works. You get to put anything that you find helpful in it. The only thing to be concerned about is if your lab notebook's this thick when you walk into the room, you're going to be spending a lot of time flipping through. But you put whatever you want in there, as long as it's not digital. No iPad tucked into your lab notebook. Solid? I know, it's a good thought. Yes, please. I think it's like 20, but it's not really a very good metric of the length of the exam, because some questions are true or false. One question is like, match the temperatures to the different steps of the PCR reaction. Some are multiple choice. Some are questions that'll take you up to 25 minutes, because it's like, here's a population, is it in Hardy-Weinberg equilibrium? Which is a short question, but you have to do a bunch of different calculations. So I think it's like 20 questions long, but it's not very, useful metric. But if you ever want to talk about what's on the lab final, folks, I'm so lonely in my dang office hours. If you want to come and talk to me about what's going to be on the lab final, I'll sit there with you and tell you exactly what's on the lab final. I'll do that. All you got to do is come to my office. All right? I've got a bucket full of candy. I've got a sink. I have a microwave oven. There's candy. Oh yeah, you were just there to not offer you any candy. I'm sorry. I have a full-size Milky Way in the bucket that nobody's taken yet. First person in my office is a full-size Milky Way in the white bucket. Keep an eye out for it. So sorry. A question, yeah.
My question was, like, the lab instructor's been taking attendance. Is that going to factor in to like the lab final or something at all, or no?
The attendance sheet is there so that basically it's used in decisions of, there will be some folks who ask to have labs reopened next week, and that decision will partially be based on, have you been attending labs? So it's factored into my choices about like how flexible we can be with reopening assignments for students, but it won't be worth actual points. It's just a good thing to know, because I'll be honest, if you've never shown up to a lab and you come to me and you ask me to open 5 labs up for you, I'm gonna say no, or maybe do one of them. It's okay, you know, it's okay to not show up to lab, but it's not okay to not show up to lab and then not do any of the work. If you're gonna not show up, you gotta like make it look like you're doing all this stuff. Anyways, of course, there's always exceptions that can be made. Life happens. Do not ever be shy. Never be shy to come talk to me and ask me for, you know, assistance or opening assignments. I don't want to discourage that behavior. Any other questions? How we doing? Okay, cool. This is expected, you know, that we eat up about like 10 to 15 minutes today, just catching up on, you know, the last little bit of the semester. Because this is when it all becomes real, people. Check your Canvas grade book. I know some people don't do that. You have so much accessibility to information that we never had when I was a student. You can see what your grade is right now. We didn't used to be able to do that. You know, we had to like take all our materials and write it on a sheet and then like calculate it on our own side. So do that. If you've got assignments that are incomplete and you're like, oh, I forgot that assignment even existed, maybe I'll open it for you. Give it a shot. It doesn't hurt to ask. Come talk to me. I want you to pass Bio 105. I do. I promise. Okay, that's the last bit. I wish I was editing these YouTube videos so I could like say skip to 15 minutes to avoid non-lecture material. But I'm not doing that. I'm not even putting full descriptions underneath these videos because that's a lot of extra work, and life hack, don't do work that you're not getting paid for. All right? That's a little secret for success. That's fine. It's just keeping us safe. Okay, folks. We finished up this PowerPoint. Thanks for watching the additional video I posted on Friday about the sex-linked characteristics. I hope it was useful. It's always weird doing a recorded lecture, but it sort of wrapped up everything in the PowerPoint that I was unable to cover Friday in person. Now, the last thing that I wanted to mention is here in polyploidy. Linkage analysis isn't really super important, but we'll talk about it briefly. Okay, so we're just going to hit the few topics that weren't included in that recording. Remember, some characteristics are encoded on the X chromosome, and males are hemizygous for the X chromosome. They've just got one copy of it. So if you express whatever allele you got from the egg involved in producing you as an organism. Okay? It's interesting. There are X-linked disorders, X-linked characteristics. I talked about them all. I talked about the hemophilia thing. Pretty good stuff. Pretty good stuff. I talked about dosage compensation as well. Linkage analysis. Okay. Sometimes genes are located on the same chromosome, so they don't follow the rule of independent assortment, and they get inherited together. That means that if you've got I'm not gonna dally on this concept. If they're on the same chromosome, they're inherited together, okay? So if the chromosome has a dominant A and a recessive B, you're likely to make a gamete that has a dominant A and a recessive B, even if the homologous chromosome had recessive A, dominant B, all right? Those are going to be inherited together. They are linked genes when they're on the same chromosome. This isn't anything you'd ever be tested on for Bio 105, but you'll do these analyses if you continue into genetics. Now, different genes that are linked on the same chromosome, all right, have what we call recombination frequencies. So if you remember synapsis and crossing over, that leads to material being exchanged between different chromosomes and generates these recombinant chromosomes. So if genes are super close to one another, they're more likely to be inherited together, because if a crossing over event occurs, they both cross over together, right? Now, that contributes to how likely genes are to be inherited together, and that's represented on these things called linkage maps, which, just to make things confusing, right? They space genes out, and the distance represents how likely they are to be separated during recombination, but it's not necessarily the same as spatial distance, even though spatial distance on the chromosome is a contributing factor to how likely recombination is. But at some point, you'll be doing analyses where you are including recombination frequencies in your math to sort of compensate for how often do these genes get inherited together and how often do they get separated during recombination. That's all. Non-disjunction events, I talked about it in the recording and in lecture, I think. Aneuploidy, monosomy, trisomy, polyploidy we didn't really talk about. In polyploidy, okay, the organism gets a complete additional set of chromosomes. So a haploid cell has one of every single chromosome. A diploid cell has two of every single chromosome. A triploid cell has three of every single chromosome. And a tetraploid cell has four of every single chromosome. And an octoploid cell has 8 of every single chromosome. All right? Plants are pretty good at tolerating full sets of chromosome replications. All right, we get an additional set of chromosomes. And you can kind of probably think about how this might arise. You just end up making a diploid gamete, and it gets fertilized by a haploid gamete, and then you get a triploid individual. And if the thing can survive, maybe it now has an additional set of backup blueprints, and it can get more characteristics crammed in there. All right? So polyploidy is very common in plants. A lot of plants have multiple sets of chromosomes. Animals don't tend to tolerate this as much. Why? I mean, I don't have a good answer for that question. So why are you all busting my chops about it? All right, great. Down syndrome, cool. We've got 3 copies of chromosome 21, trisomy 21. We talked about this, right? So let me know if you got any questions. I'll answer them. Aneuploidy of sex chromosomes. So aneuploidy, remember, is abnormal chromosome number, and it results from a non-disjunction event where you've got gametes that have extra copies of chromosomes. So sometimes you'll have a non-disjunction event and you'll make a gamete that has 2 X chromosomes in it. Or, 2 X chromosomes in it, okay? And if that happens, you make an offspring that has 2 copies of the X chromosome and a Y chromosome, all right? Sometimes you can also have individuals where they didn't receive a second sex chromosome. But as you know from chromosome inactivation, you don't usually, or dosage compensation, okay? You don't actually need 2 copies of the X chromosome to have a functioning cell. There's enough instructions to make a functioning organism in 1 X chromosome. So you can have individuals who receive only an X chromosome and no second X chromosome, no Y chromosome. And that'd be monosomy X. It leads to something called Turner syndrome, okay? It's the only viable monosomy in humans. Why? Because there's plenty of individuals with only 1 X chromosome who survive very well. I am one of them, So not having the Y chromosome doesn't necessarily lead to a ton of issues. Now, these are disorders and syndromes, right? Where individuals who have an extra X or only 1 X and no second chromosome, it usually produces non-fertile offspring. So it's sterile offspring, okay? And sometimes they'll express some intermediate phenotype, where like an XXY individuals has, the individual has many male characteristics, But anyways, when you have aneuploidy of sex chromosomes, you're more likely to have viable offspring because the sex chromosomes are regulated in their own complex way. All right. Solid. Y chromosomes. Okay. This is, I don't know why, we always talk about this in courses about like, we got Genghis, I think it's pronounced Genghis Khan, if I'm right there. Okay, and you can say, oh, you know, so many people are related to him. Okay, and you look at similarities in Y chromosomes because Y chromosomes are inherited only from the sperm cells, only from the male side. So the Y chromosome composition is entirely based on the paternal side of the parents. So you track Y chromosome compositions and you can see like, oh, what was the paternal contribution to the population. It's less fascinating than extranuclear DNA, I think. So mitochondria, chloroplasts, they've got their own genomes. I mentioned this in the recording. They reproduce independently, right? This is called extranuclear DNA. It's not in the nucleus. It's not the central genome or the core genome, but it is, they're essential for the function of these organelles. So the mitochondria, the chloroplasts, they've got their own genomes. Cool so far? Egg cells are bigger than sperm cells. Sperm cells don't, when they fuse with an egg cell, they don't contribute any mitochondria or chloroplasts. So all of the mitochondria, all of the chloroplasts that you inherit, you don't have any chloroplasts, no offense, they all came from the egg cell, all right? So from the maternal side. So that means that if your mother has a mitochondrial disorder, then you get it. And, you know, that's a bummer because mitochondrial disorders tend to lead to really low energy levels. And, you know, you don't want that. You want to be a dynamo out there in the world, just breaking down barriers all over the place. So there's this story here, which at this point older, you know, but it sure was exciting when I first heard of it back when I was a student, and so I keep mentioning it because it emphasizes a concept. So check this out. You had a couple, and the egg-producing member of the couple had a mitochondrial disorder, okay? So the mother's got a mitochondrial disorder. She doesn't want to pass it on to her child. And what are they going to do? What do you do? Okay? But the mother and the father still want to have a child whose nuclear genome is a mix of their nuclear genomes. All right? It's a reasonable enough thing to want. So what they did is they took the nucleus from the egg cell and pulled it out of the cytoplasm. So now it's just the nuclear genome of the egg cell. And then they took that egg cell nucleus and they put it into a different egg cell that had had the nucleus removed. So that new egg cell that they built had healthy mitochondria in it. Therefore, the sperm cell could fertilize it, just do IVF, right? Sperm cell fertilizes that newly constructed egg and you produce an offspring that has a nuclear genome that is a mix of the maternal and the paternal nuclear genomes, but an extranuclear genome, a mitochondrial genome, that is entirely from this donor, all right? So there was a donor for the mitochondrial genome, and you get an offspring that has 3 biological parents, okay? And this story got wild. It's totally worth looking into because there ended up being issues where the donor of the egg for the mitochondria wanted to be involved in the life of the offspring in some ways. And there was an international movement of the egg cell that made everything very, you know, out there. But that's just something to look into when you've got some time during vacation. And that's the last slide. And now we can get into the real week 13 material. And it's Wednesday, right? Sick. We're doing great then. 83 slides. Yeah, we'll do it. Okay, folks. So we've been talking about cells, we've been talking about genes, and we've never really spoken about the structure of DNA. So what does a gene actually look like? You know, like what are these base sequences of nucleotides? How are they interpreted? How are they read and turned into proteins? We talked about DNA replication occurring in S phase of interphase, but we've never really talked about what DNA replication looks like. So now we're going to actually talk about DNA, all right? We have to talk about DNA replication. It's happening all the time, and errors in DNA replication lead to all sorts of problems. So we got to talk about how it's supposed to operate. Learning objectives, you can always look at these. All right. Good. Griffith. This Griffith study is super famous. Frederick Griffith, 1928. All right. There's a bunch of there are questions about what is the molecule of inheritance. So if you remember thinking back to the Thomas Morgan experiments, where they were showing, oh, there's a chromosomal basis for sex determination, okay? It's these little chromosome things that we can see under the microscope. They can visualize them, all right? We've moved far enough now where you can see the different types of molecules. You can break the things down and find out what makes up proteins, what makes up the material inside of the nucleus, okay? And people were wondering, well, what molecule is actually responsible for inheritance? Is it DNA? Some people thought it was proteins, and proteins make a lot of sense because there's a bunch of different amino acids that can be put into a polypeptide sequence, but there's only 4 different nucleotides, okay? So there's this Griffith experiment that's conducted, and then follow-up experiments that really clarify that, oh, it's DNA, not protein. All right. We got to talk about this one. It's really straightforward. Let's just start talking about it. There are 2 varieties of this bacterium. Some of them are called S cells because they are smooth. They have a polysaccharide coat on them. Some of them are R cells for rough because they look rough when they grow a little colony. All right. The polysaccharide coat on these S cells makes them resistant to the defenses of a living organism, the immune system, right? So if you inject a bunch of these S cell type bacteria into this mouse, the mouse dies. These R cell types, okay, same species, different varieties. You inject these and the mouse is healthy. It's fine, okay? If I take the S cells and I heat them up to kill those cells, well, then I can inject those into the mouse and the mouse is still healthy. It makes sense because these cells are now dead, all right? But if you take those dead S cells and mix them up with some living R cells, so you take dead pathogenic cells and living non-pathogenic cells, okay? Well, you inject that into the mouse and the mouse dies. And when you extract the mouse's vital fluids and then culture bacteria, you find living, smooth phenotype bacteria. That indicates, okay, something moved from these S cells to these R cells and transformed them into S cells, the pathogenic type. So this is the framework experiment or the foundational experiment that shows there's something being transferred between individuals that can lead to a different phenotype, okay? So some material can be moved between and can give organisms a new expressions, new phenotypes, new capacities, okay? So Chase experiment, it's important, it lays this groundwork. Does it really prove that the molecule is DNA? Not really, because something else could be moving from the dead S cells to the R cells, all right? The experiment that confirms that it's actually DNA doing the transforming is this 52 Hershey-Chase experiment, all right? It was known that these bacteriophage viruses were injecting something into bacteria to make the bacterium produce more viruses. What they were injecting, people weren't sure yet, okay? But based on these Chase experiments, based on the Frederick Griffith experiments, people knew there was something being transferred, okay? They wanted to figure out what it was. The two main contenders, DNA and protein, all right? And they had chemically determined the structure of both of those molecules, allowing them to do this experiment. It's a little tricky. Hope you're ready. Y'all ready? All right. Here we go. Okay, I'm gonna get more dynamic here. Here we go, right? Clear some room, clear some room. Here we go. All right, so radioactively labeled isotopes allow us to track the movement of certain molecules. Hey, you know, there inside a protein, there is sulfur, okay? So I grow a bunch of viruses in the presence of radioactively labeled sulfur, and that means that those viruses that grew will have radioactive sulfur, and their proteins will be radioactively labeled, okay? So you've got these bacteria on the top that have radioactive protein because you grew them in the presence of radioactive sulfur. So when they made the sulfur-containing compounds, those compounds became radioactive. On the bottom, we've got viruses grown in the presence of radioactive phosphorus. Now, phosphorus is in the DNA, not in the proteins. Therefore, all right, these viruses have radioactive DNA. Cool? And we can track if the molecules are radioactive or not. Okay, something's being injected into the bacteria, and it's making them create more bacteria. So whatever that molecule is that's getting injected into the bacteria, that is the molecule of inheritance. That's the molecule allowing for the transformation. So here's what they do. Experiment 1, radioactive protein viruses. Put them with the bacteria, let them do their thing, okay? Then you put the thing in a little blender, you agitate it a little bit, and that knocks all of the viruses off of the bacteria, and then you centrifuge that sample. All of the bacteria go down to a pellet in the bottom, and all the virus pieces are floating in the liquid. You sample the liquid, you sample the pellet. If there's radiation in the liquid, okay, well then that means that the protein is still where the viruses were. If there's no radiation in the pellet, that means that no proteins actually got injected into the pellet. That's the key for this first experiment here. No radiation in the bacteria, which is what they found, means that no protein was being injected into the bacteria. When you repeat that same experiment with the radioactive DNA viruses, you allow them to do their thing, then knock them off of the bacteria, then centrifuge, you find that the liquid's no longer radioactive, which indicates that the viruses don't have whatever the molecule was anymore. and it's actually ended up in the bacteria. So these results here supported, oh, it's actually DNA that is the molecule of transformation, or the molecule of inheritance, or the molecule that encodes proteins. All right. So now we know, oh, it's DNA. And we know that DNA has sugar and phosphate and nucleotides in it, but we don't understand its actual three-dimensional structure. Okay? So then we've got the follow-up experiments, the Watson and Crick experiments, right? Using this image generated by Rosalind Franklin, okay? This image, you know, you see it and you interpret it and you go like, oh, that's clearly a double helical structure of DNA, no problem. I could have figured that out. Anybody could have figured that out. It's so obvious, right? So you look at that. You got Watson and Crick. Here they are, okay, looking at the 3D model. And there's so much to discuss about, like, hey, they didn't really credit her with producing that image. They were visiting the lab, and they looked at that image and used it to build their own model with that insight. It doesn't seem very fair. And yeah, probably not. This is a very famous picture. Like many famous pictures of its ilk, it is a staged photograph, okay? This is not a candid photo, so that means, I don't remember which one's Watson and which one's Crick. I get mixed. This one's Crick? This one's Crick because of the crick in his neck? Yes, I agree. It does mean that Crick here chose this expression, which is very interesting, all right? I don't know, this is the reason that I got into science, was to mock scientists and how they look. I do that a lot in my classes, I'm just realizing. That's probably says something about me. I should have played sports. I should have done that. All right, so we'll assume this is Crick and this is Watson. Could be wrong, but we got a 50-50 chance. I'd like to ask this, because when I was a student, the instructor was like, hey, anybody who can tell me what this is, I'll give you an extra credit point, all right? And I like to keep that alive. Can anybody tell me what Watson is using here to point at the model of the double helix? If anybody knows, I'll give you one extra credit point. Is that what you were going to say? It is, in fact, a slide rule. So stick around at the end of your semester. You had that too? Damn, I'm so sorry. My eyes are just... So stick around at the end of the class, and everybody else, jump this student outside. Yeah, I'll give you a point. That's a slide rule. Who the hell uses a slide rule anymore? Nobody in my class knew what it was, and every semester, this is the first semester in my 5 years here that anybody's actually known. Are they making you use slide rules again in class? No? Architecture students use them sometimes? Oh, engineering students, okay. Classic. Interesting. Either way, the point is, this is the model. These are the two people. Boom, here we go. DNA structure. Here's our current understanding of the structure of DNA. All right? Anti-parallel nature, base pair complementarity, and semi-conservative replication. Those are like three big phrases that we're going to be using and you need to understand here. So let's just look at the dang model, refresh from biological macromolecules, whenever that was, 10 weeks ago or something like that, I think. It's been a long time since we've looked at the structure of DNA, really. So remember, DNA, it's also called it's a polynucleotide. It's a polymer made up of nucleotides, okay? Each nucleotide has a sugar group, a phosphate group, and a nitrogenous base, all right? The sugar and the phosphate are the same in each nucleotide, but the nitrogenous base is different. Okay? So you've got T's, C's, G's, and A's, adenine, cytosine, guanine, and thymine. All right? You don't have to memorize the name of the bases, or the nucleotides. You just need to know A, T, C, and G, and you'll be fine. Okay? Cool. So you've got one polynucleotide here on the left side, and another polynucleotide here on the right side, and they associate together to make the DNA molecule. So the DNA molecule, it's a double helical structure, okay? 2 strands of DNA that are complementary to one another. And when I say they're complementary, T always associates with A, G always associates with C, and that's it. And then A to T, C to G, but T to A, G to C. I remember it by just thinking about what word I could spell with A, T, C, and G, and it's at. That's the only word that I can think of that I can spell. And so therefore, A goes to T. That's my mnemonic. It might work for you. Okay. Now, each strand has a 3' end and a 5' end. That number comes from the group that's being exposed. It's coming from, technically, the carbon that the exposed group is attached to. This hydroxyl group here is attached to the 3' carbon, 1, 2, 3, of the ribose sugar. The phosphate group is attached to the 5' carbon, 1, 2, 3, 4, 5, of the ribose sugar. Okay? So the 5' end, the 3' end. DNA is anti-parallel, where one strand's 5' end is opposite the other strand's 3' end. But I want to emphasize in anti-parallel, that does not mean that each strand is identical. This slide can kind of screw you up, okay, because of how this image is generated. It looks like, oh look, TGCA, and then this other strand goes TGCA. Cool. So anti-parallel means that the sequence on this strand is the same as the sequence on that strand. They're just